Welcome. It is so great to have you here. Tonight, we're going to be talking about feedback and homeostasis. And we'll be answering a couple of key questions. First of all, what is homeostasis? This incredibly important process that's fundamental to life. How do feedback loops work? Feedback loops are how homeostasis is maintained, and we'll delve deeply into that topic. How is body temperature regulated? That's an illustrative example that we'll use to explore homeostasis and feedback loops. And then we'll use another illustrative example, particularly important for AP biology, which is how is blood sugar regulated, which leads us to the topic of diabetes, type 1, type 2, and what causes it. That's what you're going to learn about tonight on the AP Bio Show. I am so happy to have you here. My name is on tonight's show, in addition to the preceding topics, we'll have a weekly quiz and we'll have tips for learning the greatest topic of all, biology. Once again, I want to welcome those of you who are watching live. It really means a lot to me that you're watching today. I want to introduce myself. My name is Glenn Wokenfeld. I'm also known as Mr. W, and I'm a retired AP biology teacher. And my mission is helping you succeed in AP, IB, or college biology. I do that through this show, the AP Bio Show. And I also do that through two things that I'll tell you more about: my website, learn-biology.com, and my app, Biomania. Before we get deeply into the show, I want to tell you that I know that many of you are studying for an upcoming test in AP biology or IB or college biology, and it'll be really useful to you if you download my ultimate AP bio study checklist. It's a list of the topics that you need to know. You can use it for self-assessment, in addition to all of the materials that I've put together for you on learn-biology.com. The way you download the ultimate AP Bio study checklist is by going to apbiosuccess.com/checklist. Let's start. We're going to begin by looking at homeostasis and two broad categories of organisms in relationship to homeostasis, and those are conformers and regulators. So, what is homeostasis? Homeostasis is the ability of a living system to maintain its internal conditions at a relatively constant and optimal level. What are a few homeostatic variables? I'll talk about ones that pertain to us, but they also pertain to many other organisms. Body temperature. You keep your body temperature at a relatively constant 37 degrees C, 98.6 for those of you in the United States. Your blood glucose level is kept in a relatively narrow range, despite the fact that you might have recently eaten or gone hours without eating. Your blood pH is kept within a relatively narrow range, as is your blood pressure, your blood calcium level, your oxygen and carbon dioxide levels, and your water and electrolyte balance. And the various systems of your body have all evolved in order to maintain those various variables. So, for example, your kidneys are really important for water and electrolyte balance, your digestive system, and systems that we'll talk about involving the pancreas and the liver are very important for maintaining blood glucose level. Those details will be dealt with later on in this show. In terms of homeostasis, there are two broad categories of animals, and those are regulators and conformers. And actually, let me say this: it's not just two broad categories of animals, there are two broad categories of organisms. Mammals and birds, to use the example of body temperature, are regulators. And what that means is that for that homeostatic variable of body temperature, they keep the temperature within a relatively narrow range. They maintain it. But there are many other organisms that don't. Those that do are called endotherms, and they're pretty much limited, not exclusively, to birds, mammals. There's a couple of species of fish and a couple of very specialized species of reptiles that are endothermic, that can maintain their temperature from within. And then there are others, and that includes a long list of organisms: reptiles, amphibians, fish, most invertebrates, plants, fungi, protists, bacteria. All the archaea are body temperature conformers. And what that means is that they let their body temperatures fluctuate based on the environment. So notice this line over here in blue. That shows that as the ambient temperature rises, the body temperature also rises. And that's true of any ectothermic organism like a snake. As it gets warmer, its body temperature increases. And in contrast to that, if you look at this example of a wolf over here, which is a mammal, note that its body temperature stays at a constant 37 degrees C, no matter what the ambient temperature is. And that's because it's regulating its body temperature. I want to say to those of you who have just chatted in that I really appreciate the comments. And I also want to say that while I have a lesson to teach, I'm completely open to questions, comments, so on and so forth. So, John Doe, thanks a lot. I really appreciate the appreciation. So, let's talk about some animals that are conformers. Jellyfish and mussels are conformers for osmolarity. And since they're oceanic organisms, what that means is that as the salinity of the water increases or decreases, they allow their body to they allow their body tissues to match that osmolarity. That's particularly important for a mussel. That's an intertidal organism. And as the sun beats down on those mussels as they're exposed to the tides, they would need to allow their salinity level to go up as the salinity goes up because the water's been evaporating. Crabs and eels are somewhat astonishingly conformers for oxygen level. So, as the oxygen level of their environment goes up or down, they allow the oxygen level in their tissues to go up or down. Other environmental variables for conformers include pH, ion concentration, and nitrogenous waste levels. But again, I want to note that mammals like us aren't conformers for almost any of those variables. We're regulators. That is going to lead us to the topic of feedback. I'm going to change our view so that you can have a better view of the screen. To do that, I'm going to go like this. There we go. Okay. Okay, let's talk about mechanisms. Let's talk about feedback mechanisms and how they connect to homeostasis. Here's an example of feedback. It's when the output of a system is also the input. So here's a system. That system could be your excretory system. It could be your entire body. Inputs are what comes in. Let's say, for example, that is food and water. Well, the outputs in this case feed back to the system. So, like, for example, here's an example for your kidneys. The input is blood. The system is the kidneys, and what it's doing is it's actually taking the blood and it's adjusting the osmolarity of the blood. That feeds back to the system in a way that tells the system how the system's been doing. So the systems in your body, as we'll see, will turn on and off in response to feedback. Negative feedback allows organisms to maintain homeostasis as they respond to internal and external changes. That's negative feedback. Positive feedback can accelerate internal changes and drive a process forward, but they both involve the output of the system feeding back into the system and the system using that to adjust its activity. I'm now going to talk about set points and negative feedback, and I'm not going to do it in relationship to biological systems. I'm going to do it in relationship to something that's a little bit more accessible and more understandable, and that is a home heating system. So, a set point is where you would set your thermostat. Let's say that you want your home to be 68 degrees Fahrenheit. So here's the set point. That's what you adjust And you say, okay, I want it to be at 68. Negative feedback then follows. So when it's 68 degrees and it's 66 in your home, then what'll happen? There'll be a sensor that'll measure the fact that, oh, the temperature is lower than the set point. And what that will do is that will send a signal to the furnace, and the furnace will go on and it will pump out heat. Well, that heat is then going to feed back to the sensor, which is the thermostat. And as there's more heat produced, the temperature will rise until it reaches the set point. And when it gets to 68 degrees, your thermostat will shut off the furnace. That's a beautiful and hopefully a very accessible example of negative feedback. Here are some biological examples, and you'll note that they're very similar to the examples of homeostatic variables that regulators control that I opened up the show with. So, body temperature in humans, 37 degrees C is the set point. Blood glucose can range between about 70 to 100 milligrams per deciliter. Blood carbon dioxide levels, their partial pressure can range between 35 to 45 millimeters of mercury. Now, homeostasis is often controlled by paired antagonistic negative feedback loops. And if you live in many parts of the United States, you won't only have a heater, you'll also have an air conditioner. So you have one system for heating the home, you have another system for cooling the home, and they'll work together to maintain home homeostasis, maintain the temperature of your home at a constant level. So one negative feedback system will respond when conditions are above the set point. So here we have our set point again at 68 degrees. And here we have a system that will turn on the air conditioner to pump out. It's actually called cool, but what we're doing is we're removing heat from the environment. That will feed back to the sensor. And when the temperature drops to 68 degrees, then the system, the air conditioning system, will turn off. And we've already talked about how if the temperature goes below 68 degrees, drops to 66, then there'll be a signal that'll go to the furnace, that'll output heat, and that'll feed back to the system. So we have two antagonistic systems that one that work to either heat the home or cool the home, and working together, they can maintain the home's temperature at a constant level. Second system responds when conditions are below the set point. Now we can really look at thermoregulation and how biological organisms do it now that we've looked at the example of a home heating system. So, thermoregulation is mostly carried out by endotherms. So, we've talked about how there are regulators and there are conformers. Well, the conformers are the ones who don't really regulate body temperature because they allow their body temperature to fluctuate with the environmental temperature. But the regulators, like us mammals, also birds, and a couple of organisms I'll introduce you to, they work, and sometimes they work quite hard to keep their body temperature at a constant level. Here is some of the endotherms that are worth knowing about. Mammals are endotherms, birds are endotherms. Then there are organisms that are partial endotherms. There's a fish called an opa, which is capable of regulating its body temperature. Tuna are partially capable of regulating their temperature, and often what will be found is that tuna that are swimming in the ocean have a body temperature that's significantly above the water that they're swimming in. And that's partly because of metabolic heat from their bodies that's enabling them to raise their body temperature and really perform at a level that would be much higher than a fish whose metabolism was completely determined by its environment. Leatherback sea turtles are also partially capable of regulating their temperature, and somewhat astonishingly, bees are as well. Bees can beat their wings without flying. They can also kind of do a sort of collective shivering within the hive, and that can significantly raise or cool the temperature of a beehive. Dinosaurs, for those of you who are interested in dinosaurs, as I am, were probably endothermic. And we'll see that they have some adaptations, including feathers, that were for insulation that help them keep their body temperature within a narrow range. So, how do endotherms maintain their body temperature? Well, first of all, there is insulation in the form of fur and fat. Fur traps air, and that layer of trapped air enables an organism to warm itself so that it won't be losing so much of its body temperature to the external environment. Whales, which don't have hair, have thick layers of blubber that enable them to maintain their body temperature in some of the coldest ocean waters on Earth. Another mechanism used by endotherms, this one for cooling, is evaporative cooling. And that involves sweat glands that are underneath the skin, and they release sweat, mostly water, onto the skin. And as that water evaporates, each one of those molecules that jumps away is carrying away a significant amount of heat. And as that does, it lowers the overall temperature of the body. Temperature is average kinetic energy. So as these water molecules become gas and carry away their heat, they lower the overall temperature of the body. There are a variety of thermoregulatory adaptations that are related to surface area, which is really cool because now that we're in Unit 4, we can learn some of the material that we learned about. We can use some of the material that we learned about back in Unit 2 of AP Biology. So one of them is going to seem a little bit counterintuitive based on what I'm showing. With cold stress, there's selection against small size. This is particularly relevant to the marine mammals. The reason is that as you get smaller, right, you have more surface area relative to your volume. And with more surface area, you can more easily radiate heat out to the environment. That is a bad thing if you're a marine mammal, that's a body temperature regulator living in cold water. And on account of that, otters are about as small as you can be and still be a mammal living in the water. And if this is a little bit confusing, sort of think of the opposite argument. There are no mice sized mammals that have adapted to living in the water. Why? Because mice are too small. Their surface area to volume ratio is too high. They would lose too much heat. They'd become hypothermic. So their selection against small size, and otters, which are the smallest marine mammals, have partly adapted by having these luxurious coats of fur that unfortunately made them targets for hunters, and they were almost hunted to extinction, but they still live in my great state of California. There's also another way that adaptation works in terms of surface area. Well, cold stress leads to selection for large size because as you get bigger, your ratio of surface area to volume goes down. So the marine mammals that have evolved best and most completely to live in water, which are the whales, are huge. Why? Simply because by being very big, they have relatively little surface area relative to their volume, and that keeps them from losing too much heat. Now, think about another aspect of this. The largest mammals are elephants. They're huge. And as being huge, they have relatively little surface area compared to their volume. But for them, that's a problem. Why is that a problem? Because they live in a hot environment and they need to be able to radiate heat into the environment. So elephants have evolved huge ears, which essentially act as radiators. Blood is pumped into the ears, and then the blood diffuses its temperature into the skin, and that in turn diffuses into the environment. So this is the thermoregulatory adaptation by elephants. So even though they're big, they increase their surface area through having gigantic ears. Here's another kind of adaptation. One is called vasodilation and one is called vasoconstriction. Vasodilation, which is shown on the left, involves muscles that surround the capillaries. Those are the smallest capillaries relaxing. And that allows the capillaries to expand and it allows more blood to flow into those capillaries. That allows more heat to be released through the nearby skin. And that's a thermoregulatory adaptation for releasing heat. When your skin becomes flushed, that's because you're vasodilated. That often happens on a hot day. Why? Because that is one of the ways that you are regulating your body temperature. The counterpart of that is vasoconstriction. And here we see constricted capillaries, and it's the opposite thing. The muscles that are around these capillaries constrict, and that reduces blood flow to the capillaries. That reduced blood flow reduces the amount of heat that can be lost to the environment. And again, it's two antagonistic systems that are working to maintain your body temperature at 37 degrees Celsius. There's another really cool adaptation that's called countercurrent exchange. That's found in birds, but it's also found in us. So the basic idea is that the arteries are the blood vessels that send blood away from the heart, and the veins return it to the heart. And the way that arteries and veins are organized in appendages like legs in birds is that the arteries are interior to and pressed against the veins. And what that means is that blood goes to the extremities. It starts out really warm because it's in the body's core, but as it goes further and further and further, it's more exposed to the environment. And the bird's feet might be many, many degrees cooler than its core temperature. But then, as the blood returns from these capillaries into a vein, what you'll see is that the vein winds up exchanging heat with the artery. So when the blood returns to the body's core, it's pretty much reached the core temperature. If you think that this is just for the birds, just think about the fact that when you're cold, your hands get cold, your feet get cold, and that's just fine. And that's because of counter current flow adaptation in you as well. But what doesn't happen is your core doesn't get cold. Your core stays at 37 degrees. There are also thermoregulatory behaviors that are seen in ectotherms. So we've talked about how ectotherms have their body temperature fluctuate with the ambient environmental temperature. So you'll see lizards doing things like basking in the sun. Why are they doing that? Because that's how they'll raise their body temperature. Because with a raised body temperature, they'll be able to have a faster metabolism, that's faster muscle contraction, and that's more effective predation. At the same time, they don't want their body temperature to get too high. So when they get too hot, they will seek shade. So those are thermoregulatory behaviors that are seen in ectotherms. And just so that you don't think that they're only for ectotherms, we are endotherms and we have tons of thermoregulatory behaviors. So, for example, when you get cold, what do? You go like this. You cross your arms against your body. What does that do? That decreases your surface area. What other thermoregulatory behaviors are there? You put on a coat. That's adding a layer of trapped air that keeps you warm. You put your hands in your pocket. That's the same thing that we were talking about before. That's the kind of adaptation that reduces surface area that's exposed to the environment. So, this is a great time for questions, comments. If you just want to say, hey, Mr. W, you're doing a fantastic job. If you want to ask me a question about anything, I'll punt and see how well I can do. But I do want to emphasize that the beautiful thing about YouTube Live is the interactive component. So if you want to ask me any questions, I'm here for you right now. Now's a great time. And if you think of it later, just go ahead and put a question in. I'll be happy to pause at a good time and answer it. So give you a minute to put those questions in. And I'm not seeing any come in, and that's just fine. So let's talk about learn-biology.com. Learn-biology.com is a website where you'll get the skills and confidence to ace your AP biology class and the AP bio exam. It's 100% aligned with the College Board's AP Bio standards, and it's been approved by the College Board as a textbook alternative. And that's why hundreds of teachers and thousands of students around the country are using it to give them the edge in AP Bio and also college and IB biology. Learn-biology.com has interactive tutorials with readings, videos, flashcards, and quizzes that teach you everything that you need to know about every topic in AP biology. Those interactions include multiple choice questions, click and drag diagrams, identify the part questions, and free response questions that are getting you ready for the FRQ portion of the AP Bio exam. You get positive feedback for correct responses, and when you answer incorrectly, you get hints and repetition. And that makes you ultimately master all the material, and you wind up with a level of confidence and mastery that is otherwise hard to get. We provide student learning guides that tell students exactly what to do, and we have a fantastic system to get you ready for the AP Bio exam with FRQs, review flashcard, multiple choice reviews, AP Bio review, click on challenges, which are kind of games, and an AP Bio speed review video that will help you get up to speed as you get ready for the AP Bio exam. For teachers, we have a kind of grade book that allows you to track the performance of your students, and there are free trials available. So risk-free, you can try learn-biology.com and see if it works for you to get you the results that you're craving. And those results are understanding biology better so that you can crush it in your course. And not only that, biology is the coolest subject of all. You want to understand it, and we can provide you with a way. Now let's turn to another example of homeostatic regulation, and that's blood glucose regulation. And that's a topic that shows up very, very often on the AP Bio exam. The system that we're going to talk about is a paired antagonistic system that involves two hormones, one of which is insulin, one of which is glucagon. And working together, they maintain blood glucose homeostasis within a relatively narrow range. Your blood glucose set point is about 90 milligrams per deciliter of glucose. When you, for example, have a big meal, and that can involve a lot of carbohydrate, for example, you'll be pouring a lot of glucose into your system because the carbohydrate will go into your intestines. Your intestines will break it down into monosaccharides, and the glucose will go into your blood. Your pancreas will detect that increased level of glucose, and it'll release a hormone that's called insulin. Insulin then goes all throughout your body, and in a mechanism that we'll talk about, but also talked about in our last lesson where we talked about cell communication, it'll induce fat and liver cells to absorb glucose. Fat cells will ultimately turn that glucose into fat. Liver cells will turn it into a kind of polysaccharide called glycogen. Now it's hours later. You've gone a while without having a meal. What'll happen is that your blood glucose will fall below the set point. There are other cells in your pancreas that will detect that, and they'll release a different hormone, a hormone that's called glucagon. Glucagon will go to the liver and to muscle tissue, and it'll direct that tissue to take stored glycogen, that polysaccharide, and break it down and release glucose. Glucose will go into the blood, and then your blood glucose set point will be restored. That'll feed back to the pancreas, and it will then take a break and stop releasing glucagon. So that's the basic system by which blood glucose homeostasis is managed within your body. So here's a little bit of a quiz question, and we've got enough people to make this fun. Which of the hormones shown in this graph is glucagon and which is insulin? And just to show you what's going on, here is your blood glucose concentration. And we talked about how the set point is at about 90. Well, notice it over here, it's dropping quite a bit. So the answer is just a one or a two. There's a drop in blood glucose, and then there's a spike in blood glucose, and that's probably associated with a meal. So the question for those of you who are watching is, which hormone is glucagon? Which hormone would um, respond to a drop in blood sugar level by increasing? So, do we have any takers for this question? Well, I'm not seeing it, so I'm going to go ahead and tell you the answer. Hormone one is insulin. How do we know? Because as the blood sugar level goes up, so does the level of this hormone. And remember, insulin's job is to get your liver and muscle cells to pull glucose out of the blood and to bring it back to the set point. We know that hormone 2 is glucagon. And why do we know that? We know that because when blood glucose was falling, blood glucagon level was going up. And then when blood glucose was rising, blood glucagon level was going down. This is a perfect kind of graph that you would see on a multiple choice question. So learn this well. Let's talk a little bit about the underlying anatomy. Not necessarily important for the AP bio exam, but cool to know about. So here's your abdomen. You've got your stomach, you've got your liver over here, and then kind of nested behind your stomach is your pancreas. It's a big mass of glandular tissue. If you took a cross section and then looked at your pancreatic tissue with a microscope, you'd find that there are all kinds of specialized cells, but the only ones that we care about are beta cells and alpha cells. They're in a kind of tissue that's called the islet of Langerhans, and these are the cells that produce insulin and glucagon. Beta cells secrete insulin, which lowers blood glucose. Alpha cells secrete glucagon, which increases blood glucose. So let's look at how insulin works. And what's really cool here is that because you've already studied cell communication, you should be able to follow this. And again, these kinds of diagrams are all over AP bio exams. And we'll look at a couple of them as we understand how blood glucose is controlled. So here's the system. It's a negative feedback system, and high blood glucose levels lead the pancreas to release insulin, which we're representing by this triangle. The insulin binds at an insulin receptor that is going to be in the cells of the liver. When the insulin binds with the receptor, it sets off a signaling cascade. And that signaling cascade travels through the cytoplasm until it reaches a glucose transporter. So, what happens is that this glucose transporter is normally closed. But when insulin binds with the receptor, the transporter opens up and it allows the glucose that's in the blood to diffuse into the liver cells where it'll be converted into glycogen, a polysaccharide, or it'll be broken down into smaller pieces. And I'm just noticing that this is one, two, three, four, five, six. This is three carbons over here. So this is probably pyruvic acid. And then finally, these can be reconstituted into fat molecules. I want to emphasize that this kind of transport over here is facilitated diffusion. We're not pumping the glucose through. We're just allowing it to diffuse through. And that's using some of our information from Unit 2 to understand a topic in Unit 4. I'm seeing a question from Yusuf, and I really appreciate questions. So thank you very much for asking. AP Bio Unit 3 hard? Yes. AP Bio Unit 3 is a very challenging unit, but you know AP Bio, you're taking it. It's one challenging topic after the next. What I suggest you do, Yusuf, is go to learn-biology.com, and I've got tutorials about every topic in Unit 3 of AP Biology. So that includes enzymes, that includes photosynthesis, and that includes cellular respiration. But I want to emphasize that while it's hard, it's also fantastic. Another thing, Yusuf, is that if you look at some of the previous episodes of the AP Bio Show, I've got episodes that talk about cellular respiration, enzymes, and photosynthesis. So if you're not getting what you need in your class, you can get them through the AP Bio Show and through learn-biology.com. And I wish you the best in learning this stuff because it's fabulous. It's so interesting to understand how cells work and how your body works. So we could play around with this a little bit, and that's what you do on learn-biology.com. So for example, I would ask you a question like, which letter indicates facilitated diffusion? And what you would do is you would type in letters A through J, one of those letters, and maybe someone's willing to do that right now. What letter indicates facilitated diffusion? I'm not seeing anything, but facilitated diffusion is being represented by letter E over here. So here's another quiz question. Which letter indicates glycogen in this diagram? And what you'd have to think is, okay, I know from unit 1 that glycogen is a storage polysaccharide. The monomer is glucose. Oh, yeah, here it is. That's got to be glycogen. So again, on learn-biology.com, you get tons of practice. Let's talk about how glucagon works. And the reason why I want to review this is twofold. One is that it's showing an example of feedback, and the other is that it's a great opportunity for us to review material that we just learned earlier in Unit 4 related to cell communication. And the reason why is because glucagon works through a G protein coupled receptor system, which is what I talked about in the last AP Bio show. Letter A in this diagram is glucagon. It's going to bind with a G protein coupled receptor. Normally, or in the absence of insulin, the associated G protein is inactive. But once glucagon binds, the G protein will bind with GTP, discard its GDP, and that will enable it to interact with adenylyl cyclase. Adenylyl cyclase will convert ATP to cyclic AMP. Cyclic AMP is the second messenger that will set off a phosphorylation cascade, very simplified over here at letter G, and that will ultimately talk to the terminal enzyme, and the terminal enzyme is phosphorylase, and phosphorylase will convert glycogen into glucose. And if you're thinking like, oh, wow, that's super complicated, it doesn't matter to me, that's not correct, because every time you've gone a little bit without eating, glucagon gets released and converts your stored glycogen in your liver into glucose so that you don't become hypoglycemic. And we end with glucose secretion, with glucose leaving liver cells going into the bloodstream. If this looks a lot like the effect of adrenaline on liver cells, it's almost exactly the same mechanism. The second most prescribed drug in the United States is a drug for treating type 2 diabetes. And now that you've learned what you've learned, you can understand it. Metformin acts upon the liver to interfere with glycogen breakdown. What happens with many type 2 diabetics is that they get a spike of sugar that gets released in the early morning when they've gone several hours without eating. So metformin will act upon the liver to decrease glycogen breakdown, and that will in turn decrease glucose secretion, and that will lower blood glucose levels. It'll act in the muscle tissue, metformin will, to increase glucose absorption and to increase glycogen synthesis because your muscles also store glycogen. So, um, in, and in addition, what metformin will do is it'll increase fatty acid oxidation. And fatty acid oxidation is an alternative pathway by which your cells can make ATP um, when they're not breaking down glucose. And this is really getting us deeply into some biology. But again, the cool thing is that you should be able to understand almost everything in this diagram after the 2 or so months that you've spent learning AP biology. So, how does insulin secretion get controlled in the pancreas? Remember that the pancreas actually can read the level of glucose that's in the blood. So, what is the mechanism by which glucose can actually be controlled? Well, it works like this. If there's increased glucose in the blood, which follows a meal, then what'll happen? That glucose will enter in through a transport protein. And then what happens? It'll go to the mitochondria, it'll get broken down, and the mitochondria will release ATP. So your ATP level will go up in the cytoplasm. That ATP will then wind up closing an ATP sensitive calcium channel. So this channel is normally open. It allows, I said calcium, excuse me. That ATP will then interact with a potassium sensitive AT. I'm going to try this one more time. That ATP will then interact with a potassium channel that's sensitive to the amount of ATP. And when ATP binds with this channel, it'll close it down. That'll keep potassium from exiting the cell. That will change the amount of potassium ions that are outside the cell. And it'll cause what's called what's called a depolarization. The amount of charge across the membrane will be lower. There are certain gates that are along the membranes of the pancreatic cells, but this is found in all cells throughout the body that are sensitive to the voltage. So the decrease in voltage will cause this gate to open. This gate is very specific. It doesn't let just anything through. It only allows calcium ions to flow through. Ca with two plus signs next to it. So that calcium channel will open. And once that calcium pours into the cell, that will interact with vesicles that will finally release insulin. Why did I just drag you through that? It's because being able to understand this kind of visual representation is a skill that AP biology students need for success. It's another example of feedback mechanisms and regulation. Now that we understand how insulin and glucagon work, we can look at type 2 diabetes and type 1 diabetes. Type 2 diabetes is essentially at epidemic levels in the United States. And here's how it works. In normal metabolism in people who don't have type 2 diabetes, then what happens is that insulin binds with an insulin receptor, as we've seen. And what that does is that causes a signaling cascade that gets a glucose channel. It's called GLUT4, I believe. And once that channel opens up, glucose gets absorbed into the cell. So this is the normal mechanism of glucose entry into liver cells that we've been talking about previously. Well, blood glucose falls because the liver and muscle tissue have absorbed glucose from the bloodstream and stored it away in their cells. If you have type 2 diabetes, and that's shown at D over here, then what happens is your cells become insulin resistant. Essentially, people who have type 2 diabetes have been consuming carbohydrate over and over and over again. And at a certain point, their cells stop responding to the insulin signal. So insulin still binds at the receptor, but that does not lead to a signaling cascade. Because it doesn't lead to that signaling cascade, then the glucose channel remains closed. And because of that, blood glucose has to stay in the blood. It has to stay in the tissues. It doesn't actually enter into the cells where it can be converted into glycogen or fat. And that high level of blood glucose, which also enters into the tissues surrounding the cells, will ultimately damage organs and tissues. It'll cause kidney damage, it'll damage the capillaries, it'll cause a variety of damage that will ultimately lead to some of the secondary effects of diabetes. Type 2 diabetes, however, should be distinguished from type 1 diabetes. Again, here's the healthy situation where insulin binds with the receptor that opens the GLUT4 transporter and glucose can enter into the cells. If you have type 1 diabetes, that's also called juvenile diabetes, though it can happen in people who are older. That's what's called an autoimmune disorder. And what that means is that your own immune system, for reasons that aren't particularly well understood, will attack the insulin secreting cells in your pancreas, and they will wind up making those cells unable to secrete insulin. So, if you're a type 1 diabetic, the problem is that you can't produce your own insulin. You can supplement that through insulin injections. And that insulin, and this says something about the unity of life, that insulin used to be extracted from the pancreases of sheep, cows, other mammals that are closely enough related to us that their insulin works for us perfectly. Now, in the 1980s, when biotechnology emerged, the gene for insulin was discovered, and that gene was actually put into bacteria. And now, almost all the insulin that's used in the United States is genetically engineered human insulin that's produced by bacteria. So that's type 1 diabetes. You don't secrete insulin. And then type 2 diabetes, as we just said, you're insulin resistant. That the insulin is produced, it binds with the insulin receptor, but there's no signaling cascade, and the GLUT4 channel never opens, so the glucose stays in the blood and in the tissues. That's it for diabetes and understanding blood sugar control. And again, before I go into the next topic, I'm just wondering if anybody has any questions. And I'm not seeing anything come in. I hope everybody's understanding this well and no one's being particularly shy. But let's go on. Positive feedback is different from negative feedback because what it does is, as opposed to returning a system to homeostasis, what it does is it amplifies system activity. So, what happens is the output feeds back to the system in a way that makes the system work harder as opposed to shut down. And again, one thing that I want to emphasize is that these terms like negative feedback and positive feedback, they're really different from the way that we use them in common speech, where you'll say, oh, I got some negative feedback from my teacher after I made a class presentation. Or I got positive feedback from a teacher, and positive makes you feel good and negative makes you feel bad. This is different. Negative feedback is a good thing because it enables systems to return to homeostasis. We've seen that when negative feedback fails, as in the case of diabetes, that can be disastrous. Positive feedback isn't necessarily good either, though in the examples that we'll talk about, it does have good effects. So positive feedback amplifies system activity and drives a biological process such as childbirth to a conclusion, after which the system shuts down. So here's how positive feedback works during childbirth. You have a woman who's pregnant, and the growth of the baby causes the uterus, which is um, where the baby is growing, where the fetus is growing, it causes stretch receptors in those muscles that are making up the uterus to be activated. The stretch receptor activation speaks to nerves, and those nerves connect with the brain. And there's a part of the brain that controls a hormone that's called oxytocin. And oxytocin gets released in response to these stretch receptors in the uterus being activated. That oxytocin gets released into the bloodstream and eventually winds up reaching the uterus. It goes all over the body as hormones do, but it gets picked up by receptors in the uterus. And when it gets to those receptors, it'll induce more contraction. So that will cause those stretch receptors to be further stimulated, and they'll send more of a message to the brain, and that will lead to more stretch receptors and more oxytocin release. And ultimately, that will lead to labor and delivery of the baby. A positive feedback loop leading to childbirth by oxytocin, leading to uterine contraction, leading to more uterine contraction, leading to more oxytocin. That's how the system works. It's positive feedback. And of course, once the baby is born, then the system shuts down because the uterus isn't being stretched anymore. A similar system works in fruit ripening. And what's remarkable about fruit ripening is that the hormone that drives the system is actually a gas. That gas is ethylene. It's very, very simple. And basically, what happens is that as fruit ripens, it releases ethylene gas. That ethylene goes into the atmosphere, and the ethylene will bind with receptors in nearby fruit. And that gets those fruit to ripen, and that induces more ethylene production. Well, more ethylene production leads more fruit to ripen. So, in one area, all these fruit are ripening at the same time because of a positive feedback loop. Increased concentration of ethylene accelerates the ripening process in all fruit, and that leads to more ethylene. Just so that you know, the folks who ship bananas and apples and all other fruit have figured this out. And what they'll do is they'll ship the fruit before they become completely ripe and start to produce ethylene. And what they'll do is they'll remove ethylene gas from the storage containers and they'll pump in carbon dioxide, and that will keep the fruit from ripening. This is some incredibly cool stuff. It's complicated stuff. And I want to emphasize that there is support waiting for you. And if you need more support, just complete the tutorials that are waiting for you on learn-biology.com. We've got tutorials about everything I've taught about tonight. What we're going to move on to now is our weekly quiz. And the weekly quiz is interactive. And I hope that you are up for interacting and participating. So I'm going to grab the URL for the quiz. I'm going to paste it in here. Let's go. Okay, so here we are. We're in the Unit 4 review page and learn-biology.com. And actually, that's wrong. Here we are. We're in the Unit 4 main. Here we are. We're in the Unit 4 main menu on learn-biology.com. I'm going to scroll down to the bottom where I go to the Unit 4 review page. And that takes us to the review page, which has a review video and it's got a list of objectives. It's got flashcards. But what we're going to do is the multiple choice quiz. To make this work, I'm going to reposition myself over here. Fantastic. And we're going to start the quiz. We're going to filter for just questions that relate to feedback. There are only 4. And let's go. All right. Let me adjust the size of this. Let's see how we do. To maintain blood osmolarity at an optimal level, Humans and other animals can mobilize membrane channels called aquaporins to the cells making up the collecting ducts of nephrons, the tubules that make up the kidneys, the organs that create urine. When the collecting duct cells have many aquaporins, water leaves the collecting ducts and gets reabsorbed into the blood. As a result, only a small volume of urine leaves the body, and that urine is highly concentrated with solutes such as urea. This is AP biology. You've got a diagram and you've got an explanation, and let's see if we can figure this out. Which environmental conditions would cause a mammal's kidneys to mobilize the most aquaporins to the collecting ducts of the nephrons? And again, I want to emphasize that you're not expected to know the anatomy going into the exam. You're expected to be able to figure it out based on the evidence. You know that when aquaporins go into the membrane of these ducts, they increase the ability of water to make it through, and that pulls water out and makes the urine more concentrated. So, which environmental conditions would cause a mammal's kidneys to mobilize the most aquaporins to the collecting ducts of their nephrons? First choice, cold weather that causes significant shivering accompanied by a lack of access to water. Second choice, hot weather that causes abundant sweating accompanied by a lack of access to water. Three, third choice, hot weather that causes abundant sweating accompanied by access to water. Fourth, cold weather that causes significant shivering accompanied by access to water. So, what I'm seeing here is that the mobilization of aquaporins brings water back into the body and creates a very concentrated urine. So, what conditions would cause the body to need to do that? And I'm thinking, oh, it wouldn't be cold weather and shivering. That's not really relevant. Hot weather that causes sweating. Oh, sweating makes me lose water, accompanied by a lack of access to water. So I can't return the water to my system. The third choice, hot weather that causes abundant sweating, accompanied by access to water. Well, I wouldn't need to conserve water if I had access to water. And fourth, cold weather that causes significant shivering, accompanied by access to water. I'm thinking, I think I've identified the answer, but I'm wondering if anyone in our audience is willing to put an answer in. I'll take a little sip of water while I'm waiting, see if any answers come in. I think I gave it away a little bit when I explained it. It's got to be this second choice. And let's see. Great job. When a mammal experiences dehydration, they'll respond by mobilizing aquaporins to the collecting duct, which reabsorbs water back to the bloodstream and keeps that water within the body rather than excreting it as urine. Let's do another question. Like humans, elephants maintain a relatively constant body temperature. Which of the following actions would have the greatest effect on decreasing the body temperature of an elephant in extremely hot conditions? First choice, flapping their ears. Second choice, huddling with other elephants. Third choice, decreasing blood flow to the ears. Fourth choice, folding their ears against their body. What you've got to think about here is surface area. See if I can make this a little bit bigger. It doesn't work that well. Okay. Flapping their ears, well, that would increase wind against the ears, and the ears are for releasing heat. Huddling with other elephants, if you huddle with somebody, you're kind of decreasing surface area, decreasing blood flow to the ears. That's vasoconstriction. It would decrease heat. folding their ears against the body, also decreasing surface area. So I'm going to go with the one that kind of increases surface area exposed to the air. Excellent. The elephant's ears provided with extra surface area. Flapping the ears allows heat to diffuse from the blood in the ears into the external environment. Let's do another question. Okay. Again, this is AP Bio. We didn't learn about this system, but you're going to be asked a question about it. The diagram below shows events that help control milk production in humans. Which of the following is a correct interpretation of how the system works? So I see the hypothalamus produces something called PIH that stops the production of prolactin. Prolactin then stimulates FIL, which stops milk production. And then this FIL is secreted in milk. But then there's some kind of feedback loop where milk production leads to the nursing baby. That stimulates touch receptors in the nipple, and that inhibits the hypothalamus. So let's see what's going on here. This is an example of positive feedback because more sucking leads the pituitary to release prolactin, which leads to more milk production. See, more sucking leads touch receptors in the nipple that would stop this, and there'd be more milk production. It's stopping these two negative things. Looks pretty good. Let's look at the others. This is a positive feedback loop because sucking increases the hypothalamic secretion of PIH. That's true, which increases prolactin. That doesn't look correct, which increases milk production. I don't know. We might come back to that one. This is an example of negative feedback because sucking stimulates tactile receptors in the nipple, which have a negative effect on the hypothalamic release of PIH. And the fourth choice, this is an example of negative feedback because three inhibitory pathways are involved. Tactile receptors to the hypothalamus, PIH to prolactin, and FIL to milk production. I'm going to go with one. Could be wrong. Nice. Sucking inhibits the hypothalamus from releasing PIH. Without PIH, the pituitary releases prolactin, which leads to more milk production. More milk ingestion leads to a larger baby and a greater need for milk production. This amplification is positive feedback. Next question. The chart below depicts glucose homeostasis in mammals. If a wolf is unable to get food for many hours, the system would lead the liver to secrete glucagon. Let's do this the right way. First choice, liver to secrete glucagon that can be converted into glycogen and fat. Pancreas, second choice, pancreas to secrete glucagon so that glycogen can be hydrolyzed into glucose. Third choice, liver to secrete insulin so that glucose can be converted into glycogen and fat. Fourth choice, pancreas to secrete insulin so that glycogen and fat can be broken down into glucose. What do we got here? Liver doesn't secrete glucagon, so that's wrong. The pancreas to secrete glucagon, that's right. And then that glycogen leads to release of glucose. It's got to be the second choice. Let's try it. Nice. If the wolf goes hours without eating, its glucose will fall below the set point. In response, the pancreas secretes glucagon, causing the liver to break down glycogen into glucose, restoring blood glucose homeostasis. We've got a summary report. We did 4 questions. We got them all right. Way to go. Let's go back to our slideshow. Okay, we finished our weekly quiz. What's your plan for this week? We finished our quiz, and what's your plan for this week? Your plan is to go to learn-biology.com, AP Bio, Unit 4, Feedback and Homeostasis, and everything that we've talked about is here. It's interactive. It'll be like a tutor that's available for you 24/7. So Can't encourage you enough to make the move. Go to Learn Dash Biology. You can get a free trial, and you won't believe how much you'll learn. I am so happy that you were able to join me this week. I'm looking forward to teaching you more biology in our next episode of the AP Bio Show. See you then. Thank you so much.